we are talking about knowledge-based score function development for protein folding. So we're talking about the work of Kim Simons from 1997 and 1999, where Kim was writing a protein folding algorithm, and we talked about how there were a couple innovations. One is use of fragments, use of Monte Carlo searches, and use of a score function that had this reduced representation of the protein. So we talked about a centroid representation of the protein, where the side chain is represented by a single point, a single pseudoatom, instead of all of the points on the protein, instead of all of the atoms of the side chain. And then because of that, we need to develop a score function that's relevant for that coarse-grained representation of the side chain. So we talked about Bayesian probability, and we put a formula up there. We talked about that the probability of getting that a current particular structure is right given a sequence, which is what you want to know when you're doing protein structure prediction. You're given the sequence, you have some structure, and you want to decide what's the probability this is the right structure. And then we're going to talk about how this is decomposed using Bayes' formula. We can take probability of structure given sequence and change it to probability of sequence given structure times the probability of structure divided by the probability of the sequence. And these terms are nice because they separate out, in the first term, things that are dependent on the sequence, probability of a sequence given the structure, and second, things that are not dependent on the sequence, probability of the structure alone. And last week we talked about the sequence-independent terms, the probability of structure. We talked about like the radius of gyration, that proteins are typically compact, and for a given length, if your protein is flopping around, that's probably not the structure of a folded protein if it's a folded protein domain. And then what I wanted to talk about today was the second term, probability of sequence given structure. The sequence dependent terms in the scoring function. So we want to know the probability of the sequence given the structure. So this means we have a given structure, and it could be something like this. And you might ask, what's given a sequence, is that sequence going to fit on here? So it's a little bit like the design problem. What's the sequence that fits well on this? But we're going to have the sequence. The sequence is known. But you could imagine threading on a sequence of amino acids here, and if you end up with the hydrophilics on the inside and the hydrophobics on the outside, that would be bad, right? Or if you end up with a bunch of arginines right next to each other, so there's all this positive charge, that would be bad. Or if you have polars and non-polars right next to each other and they're not able to hydrogen bond, that would be bad. So given a structure, you could say, does a particular sequence fit on there or not? So that's what we're going to do, and we're going to try and get this into terms where we can calculate a scoring function based on known structures. So this is what's going to happen. What's the probability the sequence fits on here, given that we have this structure? And what Simons did is put this into an expansion based on the number of amino acids we look at a time. So you could look at a single amino acid, let's say a lysine sitting in the middle, and you could say, what's the probability of that lysine being dead in the center of that protein? It's probably pretty low because charges usually aren't completely buried. That would be a single, that would be looking at a single amino acid and where it sits in the structure. The other thing we could look at is pairs of amino acids. We could look at, maybe we have a serine here, and maybe we have an asparagine here, and maybe they hydrogen bond. And the two are next to each other, and so that might be a favorable pair to be next to each other. Or maybe you have a valine and a tyrosine, and so we have two hydrophobics, and they might be close to each other. So we could also look at, do the sequences show up in pairs on the structure where it might be favorable? And then we could look for even more. We could look for three of a kind. We could put, say, an isoleucine next to there and look at the three-mers. So that's what how Simons decided to stretch out this energy term. Decided to look at first the probability of a single amino acid, so amino acid I, being in a given environment in the protein, and then multiplied each of those probabilities by each other. So what we're doing here is we're assuming each position is independent, and we look at position 1, what's the probability that the particular amino acid is there, what's the probability at position 2 that this N is here, what's the probability at position 3 that an E is there, etc. So we multiply those together, and we assume we just take a product of each individual amino acid. Then we can multiply this by all of the pairs, so we can take each amino acid I, each ij pair, so all the i's less than j, and j across all the positions, of the probability of seeing amino acid I and amino acid j given the environment. And now we're going to need to normalize this by saying, normalize by the probability of seeing amino acid I in that environment, and the probability of seeing amino acid J in that environment. So that pair term, so this is a single amino acid term, and then we have a pair term normalized by seeing the probabilities of the individual amino acids. And you can imagine we can go on with a three-way term with amino acid I, amino acid J, amino acid K, given the environment. And again, normalize now by the pairs, amino acid I, amino acid J, probably amino acid J, amino acid K, probably amino acid I, J, J, K, K, I. Okay, so this is a little bit like say a Taylor series expansion, or some kind of expansion where you're taking single terms, double terms, triple terms, or say like a Taylor expansion around a function to approximate a function. So statistically, you can make this expansion and eventually get out to an exact expression. And but what we're going to do is truncate this expression and take this first term, we're going to call probability of the environment. We're going to start with one there, and then the second one we're going to call probability for each pair. And we're going to take those two terms, and then we're going to ignore three-body and up contributions. So that could go on forever, but we'll truncate that at 2, and we'll just look at 2 terms. We can look at this probability of the environment for each term, each amino acid I, and then we can multiply them all together to get a total probability for the whole environment term, or an energy for all of the environments, and then we can do the same thing for the pairs. So, actually, let me do this. Let's take that out here and say Pn is going to be the product over each amino acid probability amino acid I given the environment. And now what we're going to call the environment is going to be simply, well, from our chemical intuition on protein folding, we expect the hydrophobics to be buried and the hydrophilics to be exposed. So what we're going to look at the environment is just how much burial there is. At each position, how buried is it? So the environment is going to be defined as the number of neighbors. This could be within, say, 6 angstroms or 10 angstroms. We could look at the structure and say how many amino acids are within 10 angstroms. And if there's a lot, then that means this is a buried position, and if it's something up on the surface, then If you have a smaller number, that would be an exposed position. Jill, ask you.
This definition is true for both terms, the environment and the pair term?
We'll come to the pair in a second. All right, so that's what the environment is, and then what's nice about doing it this way is I can look in the protein data bank and I could say, if I look at all the buried positions, every position where there's 20 amino acids within a 10 angstrom radius, I can look at what are the frequencies of seeing each different amino acid. In a buried position, how often do you see alanine, cysteine, asparagine, glutamic, all down the list, and you can get those probabilities, right? You can get that directly out of the protein data bank. So that's what's great about this formulation is we can pull that data really easily. So the probability of amino acid I, given the number of neighbors, is simply the number of amino acids of type I and that many number of neighbors divided by the total number of amino acids with that number of neighbors. Or the number of a particular amino acid divided by the number total. Okay, so you guys have done this in the homework, right? Where you've done a little bit of statistics of propensities, and you can imagine doing this. You get a high resolution set of structures from the protein data bank. You can take each position, count how many neighbors, and count which amino acid is, and make a quick dictionary with these statistics, count them all up, count up all the alanines, cysteines, asparagines, etc., get the total number, and then get these fractions. All these fractions would then add up to 1 if you sum over the amino acids. Right? So this is really easy to get. And what's great about that is it then tells us the energy of the environment to bury that particular residue at this level of burial. Okay, cool. Okay, so then the pair is done very similarly. So probability, again, I had that wrong here. So the pair probability is going to be this probability of amino acid I and the amino acid J, given a certain level of burial, these pairs together, and then, actually, let me rewrite this. Pair equals all pairs I less than J. Probability amino acid I. Amino acid J given the environment of I, the environment of J, and Rij. So that here the structure, the way we're defining the structure, or X in this case, will be the environment of the two, how buried are they, and how far apart are they. And then we're going to normalize that by the probability of seeing amino acid I in its environment and at some distance from some other amino acid, but not necessarily J. The probability of seeing amino acid J buried and at some distance from some other amino acid in the structure. Okay, so this one is then going to be the number of amino acids. If we count up the IJ pairs, and normalize that by the total number of pairs that we have. And then on the bottom we count up the number of pairs of I with anything divided by the total number of pairs, and the number of pairs of J with anything divided by the total number of pairs. Those are the fractions we're looking for. So this is counts of everything at distance Rij. We'll probably bin that, do that between, say, 3.5 and 3.6 angstroms, divided by all of the interactions between those. So these are two particular amino acids. These are all the amino acids pairs. This might be alanine to cysteine, and this might be any amino acid to any amino acid, here's alanine to anything, and here's any cysteine to anything. Okay. We can get rid of the NXX here, and we get NIJ and XX over NIX and JX. All right, is that clear? Are there questions about this term represented? Okay, so what do these represent? I mean, they're really just statistics, and we're just looking at statistically a statistical probability of, is it like a protein structure? But this does have some relevance to reality, right? So this one, the environment term, is going to tell us something about burial, meaning it's going to be capturing this hydrophobic force or this force of solvations. This one in pairs is going to say if you have two things that are so far apart, what are they likely to be? That's going to tell you about the specific interactions. So this will tell you about H bonding and specific shape complementarity. Now, is it going to tell you that valine and isoleucine like to be next to each other inside the protein? Okay, which of these two terms is going to put valine and isoleucine next to each other inside the protein?
P-pair.
Hi. So the hydrophobics, the environment term is going to drive hydrophobics to be buried. So it turns out if you just bury the hydrophobics, you're automatically going to get isoleucines and valines close to each other because you're going to bury those. One of the tricks here in normalizing, we've normalized by the probability of seeing valine buried or isoleucine buried. It takes that out of the equation. So what you're going to get here is the next higher order term of, given that you have these hydrophobics buried, are they fitting together? So this is a little different than in chemistry, you might do a G of R where you just plot how many of these are near each other. This isn't quite that because it's normalized by how many are actually buried. So once you account for the fact that there's a lot of valine and a lot of isoleucine already buried, this is the next higher order term of do they really fit together at this distance. Okay, so I think that's actually one of the reasons this energy function worked really well is because it separated this, just get the hydrophobics together and then get the shape right after that, or get the hydrogen bonding patterns and the specific interactions. Electrostatic complementarity will also go up here. So salt bridges, positive and negative charges that should be near each other also come out of that pair term. What else is unique about this formulation? So David Shortle, who's down at the medical school, has a nice article about probabilities versus propensities. And he talks about the fact that we're looking at probabilities of amino acid given the structure and not the other way around. One of the ways he interprets this is this is what evolution does. Evolution starts with a sequence and then it swaps out the different amino acids. So evolution is looking at, you have a structure for a particular enzyme and then it swaps out amino acids, and so it's dealing with, by random chance, you can replace a particular amino acid at that place in the structure, or you could, evolution can replace a pair together, and it's those probabilities that evolution is really working on. Evolution it will also move the structure a little bit, but it's a walk through sequence space, not a walk through structure space when you think about the functional. Weighting energy terms. So if you have independent features in statistics, and field of statistics are independent statistical features, an overall probability will be the probability of event A times the probability of event B times the probability of event C, right? That will give us a total probability. 50% chance of getting heads, 1 in 6 chance of rolling a 3 on a dice, and I don't know, something else for maybe 1 in 6 for a second 3 on another pair of dice. Let's see, we can, if those are independent events, then you can just multiply them together. If they're dependent events, say solvation and hydrogen bonding that are related, then maybe these things aren't quite independent and they're related to each other. And so one of the things we can do to kind of account for the fact that all of these terms might not be completely independent is we place weights on each of these terms. So these weights allow for overcounting in case some things are showing up in multiple terms. We're trying to separate it out, but we recognize we don't. We also know we might not have covered everything, so this might be a way of accounting for some of these. So each of these WI are greater than 0, typically around 1. We change this into an energy or a score by taking the opposite natural log of the probability. And if we do that to the total probability, then this, if we have the opposite natural log of the product, PA, PB, PC, weightings WA, WB, and WC, we can take the natural log and we can separate out the product into a sum. So we can have opposite let's see, opposite natural log PA, and then I can pull out the weight of A, times WB, natural log PB, opposite WC, natural log PC. And then if I let a score for each individual term, say score of A, be the opposite natural log of probability A, and same for score B, and same for score C, then I can make this weight A times score A plus weight B times score B plus weight C times score C. So by putting these weights, I can end up with linear weights on each of these score terms, and each of these score terms are the opposite natural log of an individual probability. Okay, so this is our kind of an explanation of what we're doing when we reweight some of the terms in the statistical energy function. This is what we call funnel plots. So we generate many different structures of proteins, say 1,000 different structures, and then we're going to plot the energy versus the RMSD, or let's not say energy, let's say the score. We're trying to emulate the energy, and in a real biophysical protein, we're looking for the low energy configuration. So this RMSD, this is the root mean squared distance, which I think we've done in pi-mole, to the native structure. So we would do this on a sequence we know the structure of to test, are we getting close? So if RMSD is 0, that would mean we perfectly predict exactly the crystal structure. And then as RMSD gets higher, we go further away. So what we want to see is we generate a lot of structures, and each structure will have a score. So if I have a structure that's 3 angstroms away from native, it might have a particular score. And I generate a bunch of these, and if the statistics are just random, we'll just get a cloud. But what I want to see happen is as we get closer to the native structure, we want to see that the score or the energy gets lower and lower. So that would be a successful funnel plot where the lowest scoring structures are close to native. Okay, so lowest scoring structures are close to native. And the reason is we're looking for the low free energy should be the native state. Okay, so this is not correlation. We're not looking for a line here. This is not the data correlate and energy goes up with RMSD. It's because there are many structures out here, and some might be high energy and low energy. away from the native state, we don't know anything. All we know is that the lowest should be near native, and near native is 0. And a bad funnel might be, here's RMSD versus score, and we get a big random cloud, but then maybe the lowest energy structures are out at RMSD 8 or something higher. Sometimes you see multiple funnels. Sometimes you see second low energy structure maybe at 10, but in any case they're not near 0. So this would be a score function where the lowest energy seems to be not the native. So either maybe something's goofy with the native, which is probably wrong because it's an experiment, a crystal structure, or something's wrong with the energy function. We're missing something. For some reason the native structures score high and non-native structures score low. Okay, so the decoy ranking of the near native structures. So one thing you could do is take a cutoff and say, within 2 angstroms is near native, and you can look at where that structure is, and it's obviously not the lowest. Then you can just count 1, 2, 3, 4, 5, and this one's quite far up the list. So if you were blind and you didn't know the RMSD, and you just took the lowest energy structure and you told people, I think it's this structure, they'd tell you, no, that's wrong, and they'd say, well, how about this one? No, that's wrong. How about this one? No, that's wrong. And you'd go up hundreds of decoys before you'd find this. So that's kind of embarrassing. Whereas here, the lowest rank structure, your number 1 structure, is near native. So the rank here would be 1, the rank there would be very high. So one of the measures you see in papers is what's the rank of your first near native structure. Let's see, number 3. Angstroms is a good decoy, so maybe we call this good. And here's the average score here is there. The average score of all decoys is up here, so this might be average score all. Here's the average score of the good. And then you can see the score is lower, so that's what we want. But we're going to normalize this by the standard deviation. Sigma all decoys, so we look at the standard deviation here, and it's basically saying how many standard deviations below the mean do you find the good decoys. That's what a z-score is in general in statistics. It's how many standard deviations below the mean you show up. So this is a general score minus an average over the standard deviation. So typically if you see that greater than 1 standard deviation, that's good. The more the better. What's called an enrichment. What we want to look for here is, again, thinking about if this were a blind challenge, and we took, say, the best 10% by score, does that enrich the population for near native decoys? So here we do top 10%, and you want to know, are there more decoys in the set that's in the top 10% that are near native compared to in the whole set? So enrichment is the number of decoys that are good in the top, actually typically it's more like the top 1%, 10 was easier to draw there. Number of good decoys in the top 1% divided by number of decoys in the top 1%. So here we're drawing this 1, 2, 3, 4, 5. About 4 good decoys and 2 bad decoys. So this would be about 2/3 of the decoys in the top 1% are good. Now we divide that by the number of good decoys in the whole set divided by the number of decoys in the whole set. So in the whole set, I've got here again 1, 2, 3, 4, 5, 6 good decoys and maybe 1,000 other decoys. So in this case, it'd be like 0.6% or 1% of decoys might be good in the whole set. Once we filter by score, now we've got 2/3 of the decoys are good, so we've enriched the set tremendously. Okay. So this goes back to the statistical kind of approach where you want to enrich your set in the property you're looking for. All right. So if it were random, if these points were all randomly distributed, then your enrichment would just be 1. You'd get the same fraction in the the low scoring set is in the whole set. What's good about these measures, especially in the enrichment and the z-score, is we can try different scores and we can see if things get better. So we're going to test, we'll use these measures to test each score alone. So we can take the probability, the score for the alpha-alpha interactions, the helix-helix interactions. We can calculate, we can do the probability, get a statistical score function, calculate, does it enrich for good decoys or does it not enrich for good decoys? And then we decide to keep that or not. And then we can do it also in combination, where we add all of these terms together and then we ask, does this score enrich for good decoys? And is it better when I add the helix-helix term or not? So we gradually add on different terms and see at the end which of all the terms are useful. All right, so at the end of the day, features with good z-scores in Simon's 1999, they found that ENV was great, and pair was great, the environment and pair terms, packing, meaning the bumps term and the RG, helix sheet was okay, strand terms were good, helix terms were not good, they were bad. So they ended up throwing out this helix crossing angle. Why is that? Probably because it comes out of the environment and the pair scores and other pieces like that, and there's not as much signal there. So probability of sheet, and I said packing being the bumps and the RG term. All right, so they pull all these together. And in Rosetta, this is now known as score 3. Basically because it was the third combination of things that kind of got immortalized in the code. So in your workshop today, when you load the centroid score function, it'll be called score 3, and you should get this set of scores. All right, so the summary is we use statistics to divide up different types of features that we might look for in proteins. Did a lot of statistical work on each individual feature, came up with an energy function, tested it alone, looking at z-scores and enrichments, and then put all these together and in the context of predicting whole protein structures. That's how Simons came up with their low resolution energy function. So that plus the Monte Carlo routine that we talked about on Wednesday goes together into the first protein structure prediction algorithm that was reliably successful in blind prediction challenges. So there it is.